Alright, everybody. It is 7 a.m. and I'm recording this video lecture so that you guys can learn about genetics. I believe this is the last sort of big um, content-heavy video lecture we'll have, but it's a short one, so let's just get started. So we've talked about how neurons communicate and how drugs affect that communication in the short term. So short-term effects that alter neurotransmission. But we also started to touch on long-term effects of repeated drug use and how altering neuronal communication persistently over time with drugs can actually start to change the neurons. So for example, we talked about upregulation and downregulation of receptors and transporters, or depletion of neurotransmitters, but I want to discuss how those changes happen. So remember that there are two types of receptors that sit in the outer membrane of the neuron. So here are two types of receptors, and these are called ionotropic and metabotropic. The ionotropic receptors work quickly to induce neuron firing because they pass ions, and these ions rapidly regulate the charge of the neuron, the electrical charge. So the excitatory ionic channel basically brings the charge threshold of the neuron, or the neuron, closer to its threshold for firing, for sending this electric current, and the inhibitory channels take it further away from that. And the metabotropic receptors, coupled to these G proteins inside the cell, actually goes on to interact with the ion channel. And by interacting with the ion channel, it can increase or decrease the efficacy of that channel. Remember, efficacy is the maximal effect. So both of these receptors are proteins, and all proteins start in the nucleus. Now, I don't expect you to know all of the stuff that is inside the neuron, all of this stuff, but I want you to know what the nucleus is. And the nucleus is this orange core in here, and that's located in the soma. And the nucleus contains all of the DNA. And basically every cell contains the same DNA. They just, each cell basically reads a different portion of that DNA. So I'm not a trained biologist or a chemist. My background is in psychology, so initially it was pretty tough for me to understand DNA when I first was learning about it. But this is basically how I understand it. So DNA, this is a DNA, is like a library. It's your library. And it has billions of books that basically have the instructions for how to do every little thing in your body. And you get half those books from your mom's library and half those books from your dad's library. So half of your chromosomes from one parent, half from the other. And the chromosome is basically these books on bookshelves packed really, really tightly so that they can fit inside a tiny, tiny cell. And you have books basically for every little thing in your body, including proteins. In fact, mostly proteins. And these proteins, as far as we're concerned, consist of, or are enzymes, transporters, receptors, G proteins, ion channels. So how does the instruction book from this library get from the nucleus all the way to the synapse? And I'm noticing that the titles for these slides don't really apply to what I'm talking about. Just ignore them. It's early. So how does the instructions from the library, the nucleus, get all the way to end up being a protein receptor in the synapse? Well, there are these messengers, and each messenger is responsible for one or maybe a few books in this library. And this guy's job is to basically go to the library, grab his respective book, and Xerox the right pages a bunch of times. And then he takes that copy out of the nucleus to the factory where they build the protein from their warehouse. And the warehouse is filled with amino acids. So you have all your instructions in here. You have a little messenger guy. He goes and grabs the instruction book, makes a bunch of copies, takes it to the factory. The factory makes the protein, and the protein is delivered down to the synapse. Well, when a particular receptor is being overworked, let's say you're taking, what could you take? Let's say you're taking a lot of a beta agonist, and that's going to like elevate your heart rate. So when a particular receptor, this beta receptor, is being overworked, the messenger, that guy who goes to the library, is basically told, hey man, we're getting slammed up here. Why don't you put the brakes on for a bit so this neuron doesn't get overworked? And so the messenger slows down. He stops bringing 30 copies of the book to make that receptor, and maybe he brings only 10 copies. On the other hand, the messenger may be told to speed up production. So instead of bringing 30 copies of that instruction book, he brings 50 copies to the factory. So when we talk about expression being upregulated or downregulated, we're talking about how many copies of that receptor or transporter or enzyme instruction book are brought to the factory. And that's going to increase or decrease the number of proteins that can be made. And that's basically how your DNA regulates neurotransmission over time. And so you get these long-term effects of drug use that affect neurotransmission even when the drug isn't around. So basically, how does this message, this memo, get to the messenger to tell him, how many copies to make? Well, that's done by these metabotropic receptors that we were talking about before. So in addition to having these acute effects of regulating the activity of the ion channels, which obviously it's not here, they have this longer term effect where all these intracellular signaling pathways go down into the nucleus to basically tell the messenger how much of a receptor is needed. And there are also intracellular receptors, like hormone receptors, that do the same thing. And because these hormone receptors actually live inside the cell, and they themselves travel into the nucleus to directly interact with the DNA, hormones are actually able to induce pretty quick changes in DNA expression. So there's one more thing I wanted to touch on, and that is epigenetics. So you have so much DNA that it needs to be packed really tightly to fit in the nucleus. So you can see you have all this DNA, and it basically gets smashed down to fit into this chromosome. So you can think of this as like your headphones. The cord on your headphones is your DNA strand. And if you smash up that headphone cord so that you can fit it in your pocket, there are going to be a lot of different parts of that cord that are covered up. So there'll be parts of the cord that are covered up by other parts of the cord, and conversely, there'll be parts of that cord that are visible and accessible to the outside. So DNA is the same way. It's getting smashed up, and there are parts of it that are accessible to the messenger and parts of it that aren't. So books that are in that library, keeping the library metaphor, that are really important have little molecules around them. And these little molecules are able to kind of make space in between all the smashed up DNA so that the messenger can get in there and get to the DNA, get to the book. So epigenetics is really about where these molecules are placed. And where these molecules are placed is actually quite sensitive to our environment. So changes in environmental input like drugs, stress, learning, this can change where these little helper molecules are in the library, which affects how much a particular book for a receptor, a transporter, or enzyme can be expressed. And this is what epigenetics is. And it means that changes in how your genes are expressed can occur without the DNA itself being changed. So it's always the same DNA. It's just how easily can the messenger access the gene or the book for a particular protein, a particular receptor, transporter, or enzyme. So the easiest sort of reflection of how DNA affects neurotransmission is when genetic differences induce psychiatric disorders. So instead of these changes that are induced by drugs or environment or stress, these are differences that you're born with because your genes are different. So one example is depression, this little sad guy here. And individuals with depression often have a different gene for the serotonin transporter, SERT. And that is to say that the book for the serotonin transporter has different instructions than what most people have. And this difference in instructions causes a different type of serotonin transporter to be made in the factory. So for people with depression, this particular difference causes a serotonin transporter to be made that works better than normal. So here's a typical SERT, and then here's a SERT variant with these bolder lines. And so the SERT transporter that works better than normal basically causes serotonin to be taken up more quickly, leaving less serotonin available for binding to receptors. And researchers often look for differences in gene expression in people with psychiatric disorders to try to identify markers that might indicate ahead of time that people will develop a disorder. And in the case of addiction, they've been looking for a biomarker, a genetic biomarker for predisposition to addiction. And that's it. That's the end of this lecture. I guess I don't have any notebook concept slides, but really just know that metabotropic receptors have short-term effects of altering the activity of ion channels, but also know they have long-term effects of actually altering how your DNA is expressed and how many receptors, transporters, enzymes are available to be used. And I think that's really the gist of what I want you to know.